We continue in part 3 of activity 3 genotype in Jeffrey's technique. Since you have already watched in the previous video the steps of Jeffrey's technique in details, so now we can list briefly the steps. The steps are well understood. I think that the steps are well understood. So the first step is the extraction of DNA from nucleated cells, such as hair cells, saliva, blood, mainly white blood cells, and not red blood cells, since red blood cells do not have a nucleus. After that, we cleave DNA using restriction enzymes in order to obtain DNA fragments and in order to be able to isolate the DNA sequence of interest. Then we separate the obtained DNA fragments on gel electrophoresis. Then the gel is transferred and fixed onto a nitrocellulose or nylon filter paper. This technique is called southern blotting. We blot the DNA fragments. We denature, during the blotting, we denature the DNA double-stranded DNA molecules. We separate the DNA strands in order to allow the hybridization of a radioactive probe, which is a single-stranded DNA sequence made in the lab and which is complementary to the desired DNA sequence. So the denaturation, the separation of the double-stranded DNA into single-stranded DNA molecules allow the hybridization or the fixation of the radioactive probe with the desired DNA sequence. After the hybridization of the radioactive probe, we wash the filter paper in order to eliminate excess probe. Then we can apply an X-ray film in order to visualize the hybridization of the DNA fragments with the probe by autoradiography, and we obtain the DNA pattern or the genetic printing. Now, restriction enzymes are extracted from bacteria. They cut DNA at specific sites called recognition sites. The restriction enzymes are called biological scissors. As you can see here in this photo, we have a DNA sequence having a certain sequence of nucleotides. We use an enzyme called EcoR1 extracted from a bacterium called Escherichia coli or E. coli. That's why the name of this enzyme refers to the bacterium from which the enzyme is extracted. This enzyme EcoR1 recognizes a DNA sequence of 6 nucleotides, which are in this order: G, A, T, T, C. So the enzyme cuts between G and A. So we obtain 2 fragments of different sizes. Now, for the denaturation of the double-stranded DNA, this denaturation is important in order to allow the fixation or the intercalation of the DNA radioactive probe between the 2 DNA strands. So this DNA probe will bind by complementarity of nitrogenous bases to one of the 2 separated strands, and we can visualize this hybridization by autoradiography by applying an X-ray film. So the probe is made in the lab and it is complementary to the DNA sequence we desire to found or to find. So here are the steps. I will not repeat all steps. So as you can see, the extraction of DNA, the cut using restriction enzymes, separation by electrophoresis, blotting in order to transfer the DNA bands from the gel onto a filter paper in order to visualize the bands. Then we add the probe and we can obtain a DNA pattern containing the DNA sequence we desire to find. Now we arrive to an exercise, application exercise on Jeffrey's technique. A normal couple having 3 children. One of them is affected by a hereditary disease, and the couple expects a fourth child. In order to know whether the child or the fetus is affected or not by the disease, so the doctor performs a Jeffrey's technique for all the members of this family. And concerning a gene of interest, so we obtain 2 bands, A and B, which corresponds to the 2 alleles of the gene. The thick band indicate that the individual is homozygous for the studied trait. When you see thick band, this means that this band contains 2 identical DNA fragments having same size. So the 2 fragments or the 2 alleles here are identical. When you see thin bands, means that these bands contain different alleles or different DNA fragments of different sizes. So when you have 2 bands, the individual will be heterozygous for the studied gene. When you see 1 band, this means that the individual is homozygous for this trait or for this allele. You have to answer these questions, and we correct this exercise later on. You have to describe the Jeffrey's technique represented in document 1. You have to determine according to document B, the allele responsible for the disease. You have to specify by justifying which of the 2 alleles is dominant and which one is recessive. You have to identify or specify the genotypes and the phenotypes of the parents and child one, and you have to find whether the fetus will be affected or not. Good luck and bye bye.